talk about the diversified kingdom of Archaebacteria. We will first start with the history. This slide shows the history based on 16S rRNA, that is molecular phylogenetics. It states that the microbial life was evolved at around 4 billion years ago, though the Archaebacteria was evolved later. History based on isotopic records. Isotopic means the radioactive traces. It was found that at around 2.7 billion years ago, the methanogenesis was found. Methanogenesis is the characteristic of Archaebacteria. But the presence of isoprenoid chain of membrane lipid, another uniqueness of Archaebacteria, was found at around 1.6 billion years ago. A controversy is there. History based on microfossil. What is microfossil? The Archaebacteria are extremophile. They are found in case of hydrothermal vent, that is very hot. When the temperature is getting reduced, the sinters are formed by the silica, the gelatinous amorphous silica. Within such sinters, the parts of microorganisms were embedded. From that traces, it was found that the presence of microorganism, that is mainly Archaea, was found at around 3.8 billion years ago. So this is in all Kokwe, Kenya, the microfossil. Other than this, the oldest microfossil was found in Ishwa district, West Greenland. The origin of Archaebacteria was found in case of Yellowstone National Park, USA. Let's talk about the evolutionary theory. First, by the Woos and Fox, it was proposed at around 1977. The Archaea was a part of Euryarchaeota kingdom along with bacteria. But further, they proposed it as a domain of life with bacteria and other eukaryotes. Evolution theory proposed by Woos and Fox based upon 16S rRNA states that the coevolution along with viruses and the evolution of Archaea from that of bacteria as a domain of life. By Woos, Candler and Wolf, another hypothesis was proposed. They proposed the presence of primitive ancestor from which the Eukaryotes and Archaebacteria was formed. By Kevela Smith, another hypothesis was proposed for archaeal evolution. He proposed the theory that from Eukaryotes, the Archaebacteria was found for the presence of hyperacidity as well as hyperthermophilic environment because the ancient earth was so hot. According to Ramesh Gupta, who worked with Archaea enormously, he proposed the theory of evolution of Archaea from gram-positive bacteria. He proceeded the proof by the presence of single cell wall in case of gram-positive bacteria as well as in case of Archaea. So, from gram-positive bacteria, gram-negative bacteria as well as Archaea was formed. According to different theory, it was proposed that, especially by Woos and Candler, that is from thermophilic bacteria by horizontal transfer of genes, the Archaea was evolved, right? So that means at very ancient time, Thermotoga, Equifax, this sort of thermophilic bacteria was evolved. And according to several metabolic traits and the presence of HSP70 and glutamate synthase 1, such proteins also prove the fact that from these thermogenic ancestors of bacteria, the Archaea was evolved. The Thaumarchaeota and Acufex, they are bacteria, but their relatedness with other bacteria like Bacillus satilis as well as E. coli is much more longer than that of the relatedness with that of Archaea. So it proposed the fact that by horizontal gene transfer from gram-positive bacteria by the presence of selection pressure, that is especially the antibiotic sensitivity, such sort of horizontal gene transfer help us for archaebacterial production as well as evolution. Vertical relationship means that is from primitive ancestor, the Eukaryotes and Archaebacteria was evolved, then ultimately they give rise to the eukaryote. So we all know about the 6 kingdom system, that is Protista, Monera, Plantae, Fungi and Animalia. So within the Monera, Archaea and bacteria was evolved. And by Woos and Fox, it was proposed at around 1977 that the Archaea is taken as a domain of life along with bacteria as well as eukaryote. So from this slide, the domain of Archaea is observed. And it was also stated that the Archaea and bacteria both are taken as prokaryotes, that means without proper nucleus. Phylum of Archaea. We have already talked about that the Archaea is placed as the domain of life. It has 18 to 23 phylum, but 8 are well characterized and we will talk about 5 among them. There are several aspects by which Archaebacteria resembles with prokaryotes as well as eukaryote, like the presence of operon, 70S ribosome and envelopeless nucleoid relates the Archaebacteria with other eubacterial prokaryotes. But with respect to the information processing like RNA polymerase II, the DNA damage repair, according to other function for the energy production, it is closely related to eukaryotes more than that of the prokaryotes. We will talk about the general one, the special one as well as phylum based classification. Let's first talk about the special species like classification. So you can observe here 2 organisms are there. Number 1, Ferroplasma acidophilum as well as Halorubrum luteum. Though the evolutionary theory proposed the horizontal gene transfer, but these 2 organisms do not want to share the transfer of genetic material to and fro. So they behave as species-like entity. Group based or general classification. You can observe how many groups are there, right? As I have already told you, the Archaebacteria are extremophiles. So they can be found at extreme condition. So they may be either hyperthermophile, alkaliophile, acidophile, psychrophile, methanogens, etc. Phylum based classification. So there are main 5 phylum are there in respect to the other properties. What are they? Number 1, Euryarchaeota. Number 2, Crenarchaeota. Number 3, Corearchaeota. Number 4, Nanoarchaeota. And number 5, Thaumarchaeota. At around 1998, Margules and Schwartz proposed the different classes and orders of Archaebacteria as well as Euryarchaeota. But they did not introduce thermoplasma within Euryarchaeota. But then Garrity at around 2001 and Gao and Gupta at around 2007, they have included Halobacterium, Methanogens as well as the thermoplasma within Euryarchaeota phylum. There are different types of classification among Euryarchaeota. Let's first talk about the taxonomic classification. What are they? Halobacterium species, Methanobacterium species, Methanococcus species, Archaeogloba species, Thermoplasma species, Thermococcus species. Let's talk about them individually. First one, Halobacterium species. This is the picture of Halobacterium salinerum, right? So it can withstand very high salt concentration. So to withstand very high concentration of salt, to withstand the positive charges. It has some sort of negative glycoprotein layer. Next, this is the picture of Methanobacterium bryanti. This is the picture of Methanococcus janesi. It is very interesting that the Methanococcus janesi genome was totally sequenced. This is the picture of Archaeoglobus beneficius. You know that they are closely related to the Methanogens, and we can find the presence of coenzyme M, which is unique in case of Archaeoglobus species. Thermoplasma species. This is Thermoplasma acidophilus. This is wall-less Archaea. They actually withstand very high acidic condition as well as very high temperature. This is the picture of Pyrococcus fumari. Actually, Pyrococcus fumari is hyperthermophile. It can withstand very high temperature, right? So, as it can withstand very high temperature, their enzymes are extremoenzymes. So, PFU DNA polymerase is extracted, and that is actually used for commercial purposes, for scientific purposes as well, because this polymerase is used in PCR analysis. Let's talk about the ecological and biochemical classification of Euryarchaeotes. It includes extreme halophile, methanogen, and wall-less thermoplasma. Extreme halophile. You can see the Halococcus species, and this is deep sea. We can find this sort of salt lake in case of Utah. So, extreme halophile can withstand very high salt concentration, even 10 times higher than that of the ocean salt concentration. Some of the organism can withstand 30% salt concentration, rather sodium chloride concentration. These Halobacterium species, halophile, have some sort of structural characteristics like negative glycoprotein structure, by which it can withstand such elevated salt concentrations. Methanogens. From the name, we can understand they can produce methane. So, from this picture, you can see the Methanococcus as well as Methanobacterium, and the source of such methanogens are Avinprobsea. Methanogen, so that means they can produce methane gas either from inorganic substrate, so they are called as lithotropes, right? So, they can produce methane from carbon dioxide and hydrogen like Methanobacterium, Methanococcus, etc. In case of organotropes, they can produce methane gas from formol, methanol in case of methanosarcina, and from acetate in case of methanothrix. You can find methanogens in intestinal tract of ruminants, as well as we can find the presence of such methanogens in our intestine also. We can find the presence of methanogens in muck of swamps as well as in oil degrading places. So, it will help commercially to degrade oil by producing methane gas. Among the methanogens, there are several classifications according to the extremophile. The first one, thermophilic methanogens. We can find it in hydrothermal vent. This is the Methanobacterium thermolithotrophicus. So, it can withstand very high temperature as well as it can produce the methane gas. Psychrophilic methanogens. It can withstand very low temperature at around -10 to 15 degrees centigrade. We can find them in Alaska as well as Antarctica. This is the psychrophilic methanogen. It is Methanococcoides fumari. Acidophilic methanogens. Though for methanogenesis, the neutral pH is needed, but there are some methanogens which can withstand pH at around 2. So, in Lassen National Park, some methanosarcina was found. Those are acidophiles, though they are the methanogens. Halophilic methanogen. This is the picture of Halobacterium halobium, and this is a river in Senegal, Bidba River, which is actually the pink in color. Why pink in color? Because Halobacterium halobium has bacteriorhodopsin, a pigment which is pink in color, that render pink color of the river. Alkaliphilic methanogens. Again, the methanogenesis though occur at around pH 7 or 8, but there are some alkaliphilic organism which can produce methane at around pH 10. So, this is the picture of Methanococcus thermoalkylophilous, and they are found in Soda River in California. Barophilic methanogens, which can withstand very high pressure as well as temperature. So this is the picture of Pyrococcus absci, and this is the picture of Fiji basin where actually they are found. It can withstand 20 to 50 millipascal pressure as well as 65 to 95 degrees centigrade temperature, and there are some organisms like Methanogebacterium thermolithotrophicus which can withstand high pressure like barophiles as well as which can produce the methane gas. So they are the methanogens as well as they are the barophilic methanogens. Next biochemical division of Euryarchaeota are wall-less archaea. So you can see in Solfatara field in Italy the presence of such Thermoplasma acidophilus. It does not have any proper cell wall, right, for the evolution to withstand high temperature as well as high acidic condition. It can withstand the pH from 0.5 to 0.4 and the temperature range is at around 80 to 90 degrees centigrade. The interesting thing is that they have some sort of boundary made by phosphoglucohydrocarbon and some sort of glycolipoprotein, but they lack the sterols. This sort of evolution of cell wall structure help them to be the acidophilic in nature. Crenarchaeota usually have various other types of shapes like triangular shape and natural coccoid shape, as well as they have the flagella. Usually the sulfolobus is very interesting within the Crenarchaeota. According to the taxonomic classification, we will discuss about 3: sulfolobus species, Pyrodictyum species as well as Thermoprotea species. Sulfolobus species. Actually, this sort of sulfolobus species is found in case of Lassen National Park as well as it can be found in Solfatara field in case of Naples, Italy, which is filled with the acidic environment. Sulfolobus solfataricus as well as there are other organisms like Sulfolobus acidocaldeurus. They can withstand very high acidic condition because they can oxidize. They have the property of metabolism by which from H2S and elemental sulfur they can produce their energy. That's why they can endure such acidic condition. Pyrodictyum species. It is Pyrodictyum absci and the place is the hydrothermal vent of Yellowstone National Park. Pyrodictyum species is hyperthermophile. It can withstand 400 degrees centigrade temperature even. Next one, Thermoprotea species. This is Nevada geyser where actually this Thermoprotea species is found, and again Thermoproteus is thermophilic in nature. According to biochemical characters, the Crenarchaeota may be hyperthermophiles as well as thermoacidophiles. Hyperthermophiles are such an organism which can endure very high temperature. In case of general thermophiles, the optimum growth temperature is 55 to 60 degrees centigrade. But in case of hyperthermophiles, it is 110 degrees centigrade around. You can see, so this organism is named as Pyrolobus fumari. What is the meaning? The chimney of the fire lobe. And this is the place. The place is called Black Smoker Ridge inside the Atlantic Ocean. So it can withstand very huge temperature. Thermoacidophile. That I have already discussed about the Sulfolobus species. This is the picture of Rio Tinto River, which is acidic in nature. We can find the presence of various types of Sulfolobus species within the Solfatara field as well as in Rio Tinto River. Archaeota is recently actually named because earlier it was thought maybe as a part of Crenarchaeota or maybe as a part of Euryarchaeota. The synonym is Xenarchaeota. So this is the picture of Corearchaeum cryptophyllum, and in Kamchatka we can find the presence of Corearchaeota. They have different types of geovars. Two main types are there, PGP27 as well as PGP78. They can be attached to their host according to their closely related ignicoccus. Nanoarchaeota, very small organism. From the name you can understand. Nanoarchaeum was actually found at around 2001. This is the Nanoarchaeum equitans found at around 2001, and in 2006 from Richmond mine other Nanoarchaeum organism was also found. It has very shorter genome at around 0.5 megabase and the length of the organism is at around 400 nanometer. Thaumarchaeota. Thaum means wonder, and they are actually the ammonia oxidizer. They have some sort of ammonium oxidizing monooxygenase by which it can oxidize the ammonia in extreme condition. They found in case of Lucerne, Switzerland, but interestingly earlier it was called as the mesophilic Crenarchaeota, right? But then it was named as Thaumarchaeota. Some scientists found it as the Eukaryote, but nowadays they are placed in the phylum within Archaebacteria. 4 species are there, including Nitrosomura meritimus. So this is the picture of Nitrosomura meritimus. There are Nitrosophira also. This is dendrogram analysis of different Archaebacteria, including Crenarchaeota and Euryarchaeota. Dendrogram analysis gives idea about genetic relatedness among different species as well as genus. So from this picture you can understand how closely Sulfolobus is related to Pyrodictyum and Thermoproteus and how they are related to other members of Euryarchaeota. There are so many controversy as well as confusion according to evolution of the Archaebacteria, though the main hypothesis was taken according to Woos and Fox, that is by 16S rRNA as well as horizontal gene transfer, the Archaebacteria was developed from gram-positive organism according to the selection pressure, and they have placed as a domain of life along with bacteria and eukaryota. We have already discussed the different phylum of these Archaebacteria, and they are named as Euryarchaeota, Crenarchaeota, Nanoarchaeota, Coraarchaeota and Thaumarchaeota. Different phylum was also stated according to genetic distances and genomic, morphologic as well as biochemical structures. Thank you.